Morning, folks. So while everyone's getting settled, just a couple quick announcements. So hopefully everybody was able to get onto BSpace and find the lecture notes for this week's set of 3 lectures. Those are there as PDFs. I encourage you to go there and print those out ahead of time or have a look. I also encourage everyone to read the book chapter sections that I list at the beginning of each lecture set before you come to class. I think it will make the lecture a little easier to follow. You don't have to do that, but that's a recommendation. The other thing is students are asking, what can I do to keep up with the material, learn the material? Couple things. One is I really recommend going to the back of the book and just working your way through the problems in the back of the book. For those of you that do have Mastering Biology, you can also work your way through those problems, although we don't require them. And then I will be giving out some ungraded problem sets, and those are also a good way to test yourself and to see the types of questions that I'm likely to ask on an exam. You can, of course, also work through the exam reader to see what we've asked on past in exams in past years. Okay, so last week we basically talked about the chemistry of life, and we talked about the macromolecules of life. And this week what we're going to talk about is cell structure, and we're also going to talk about the structure of the cell membrane, because that's such a fundamental part of the way that cells and organisms function. So what I want to do today is discuss the first half of chapter 6, these pages right here, and we're going to talk today about various methods that scientists use to study cells, differences between, fundamental differences between different kinds of cells. We're going to talk about protein trafficking, so how do proteins move around in cells, and we're going to talk about organelles and what those are and what they do. All right, so first of all, you probably all know that all organisms are made up of cells. And the way that we define cells is that cells are basically the simplest collection of matter that can be alive. Okay, what does it mean to be alive? Anybody know? What do we define as a living thing? Anybody? Yeah. Say again? It moves? Okay, that's one thing, but there's a really fundamental way that we define a living thing. Really fundamental. Yeah. Reproduce, right? Exactly. So some people might, sometimes people say, well, what about viruses? Aren't viruses alive? And the answer is most scientists would say they're not. And the reason is that viruses are genetic information that can get into cells. They can replicate when they're inside of cells, and they require, actually, some of the cell's machinery for replication. So most people don't consider viruses to be alive. They can replicate, but they're dependent on cells to do that. Whereas a cell is not dependent on any other entity to replicate. It just needs the basic chemical materials to do so, and then it has all the machinery in the cell to make copies of itself. All right, and so what we're going to see is that cell structure correlates to cell function. And all cells are related by their descent from earlier cells. So if you really think about that, that actually leads ultimately to a chicken and egg problem, right? How did we ever get the first cell? And the answer is nobody really knows, actually, right? So it's a fun, interesting thing to think about. There's a lot of research in this area to try to understand how the earliest cells might have been able to evolve from simpler building blocks. But the answer is we don't really know what the first cell looked like. We can make some good guesses based on very simple cellular life forms that we see in modern biology, but we don't actually know what the first cell looked like exactly. Kind of interesting thing to think about. Okay, so how do we study cells? So these are typically too small to be seen by the unaided eye, and they're quite complex. And so one of the things that was developed early on, back in the 1600s actually, was the light microscope, a way to magnify cells so that they could actually be viewed, and people could start to see what they were and what kinds of components they had, how they worked. And what we'll talk about today briefly are different kinds of microscopes. And what I also want to show you is that microscopy, which is the ability to actually visualize cells, goes very much hand in hand with another fundamental tool of biology called biochemistry, in which we're able to purify molecules from within cells and figure out what they actually do. And as you'll see, these two basic methods kind of go hand in hand. When we can look at cells and fractionate them and identify what kinds of molecules are in those cells and what they do, then we can really start to understand what the different parts of the cell are contributing to the functions of the cell, the life of the cell. And we'll talk about that as we go through this material. So microscopy is basically a way to see cells, or anything else, but in this case cells, that are too small to see with the naked eye. And as I mentioned, the very first kind of microscopy that was developed was the light microscope, in which visible light passes through a specimen, and then it goes through a lens, which will magnify the image. Now, very typically, with light microscopy, we can get a magnification of roughly 1,000-fold. That's about the best we can do with light microscopes. And there's some tricks now that allow people to maybe do a little bit better than that, but that's sort of in general. With a light microscope, you can amplify things, magnify things about 1,000-fold. And the quality of the image depends on three things. One is the magnification, right? So this is the ratio of the object's image size to its actual size, right? So as we said, with a light microscope, about 1,000-fold is what we expect to be able to do. The second thing is resolution. Now this is different than magnification. Resolution basically means the clarity of the image. In other words, how close can two objects be, and we can still resolve them as two separate things before they kind of become merged in the image. Okay, so that's resolution. And then the third thing is contrast, visible differences in different parts of the structure. And so as we'll talk about today briefly, scientists have come up with different ways to improve and increase the contrast in biological samples, because without that, it can be actually very difficult to see features of cells and tissues, because they're just too similar. It's hard to really distinguish one part from another. So this is a figure that's in your book, and it just kind of shows you very roughly sort of scale of the size of different things over here, and the way that we can expect to visualize them over here. So with the unaided eye, we can see down to roughly 50 microns or something like that, and or maybe bigger than that, probably a couple hundred. And then with the light microscope, we can see things that include plant and animal cells, and in some cases also the nucleus of the cell. We can often see bacterial cells, even though they're a lot smaller than plant and animal cells. We can see those with light microscope, and even some organelles like mitochondria, as we'll talk about, which are compartments that are inside of the cell. Now, with modern methods, as I mentioned, people have been able to extend the resolution of the light microscope a little bit further, right? So it's actually possible in some cases to see very tiny bacteria, even some viruses, which are smaller than bacteria, with the light microscope. And that's through the use of some very sophisticated ways of filtering light, of filtering out background signal, and focusing on the image and removing extraneous light that comes through that's not in focus. And so, and your book talks a little bit about that, and there's lots and lots of great information on the web if you guys are interested in how these different types of microscopy actually work. You can find tons of interesting information. There's lots of research going on right here at UC Berkeley, various faculty members that are working actively to improve the way that microscopy is done, the way it works. Also lots of work going on at the Lawrence Berkeley National Lab, which is right here, right adjacent to the campus. So you guys are in really a very exciting place where a lot of the fundamental development of microscopy is actively going on. Okay, so a little bit more about light microscopy. So light microscopes, they can magnify about 1,000-fold, that's what we said. Various techniques are available to enhance the contrast. And typically with light microscopy, the way that the contrast is enhanced is by using various kinds of dyes or stains that will differentially stain different parts of the cell. For example, we can use dyes that will preferentially stain nucleic acid in the cell, like DNA and RNA, and will not tend to interact as much with proteins or lipids. Anybody have any idea how could a stain work that way? How could it be selective like that? Yeah. If the stain has water in it, right? So that's certainly part of it, right? So you're on the right track. It's basically thinking about the chemical differences between nucleic acids and something like a lipid, right, which is very greasy. So if you had a dye that tended to be more hydrophilic than hydrophobic, it might tend to interact more with certainly the exterior parts of the nucleic acid. The interior parts would be more hydrophobic. So that's exactly right. So we try to take advantage of the chemical differences between proteins, nucleic acids, and lipids when we develop stains to allow us to differentiate between those different components in the cell. Okay. So in general, with light microscopy, that's the way that these types of chemicals work to increase contrast. And it's important to note that most subcellular structures, that means structures that are inside the cell, are actually too small to see with a light microscope in general. Okay, in general, that's true. All right. So one of the things that I do want you to come away knowing from this is, you should know roughly, what type of method would be appropriate for studying or answering a particular kind of question. Right. So you would know that a light microscope is great for looking at cells and sort of overall cell structure with appropriate stains, but not so great for looking at things that are smaller than the cell. All right, and so the next slide just shows some examples of this. So this is a kind of a cool experiment. This is actually the first science experiment that my son did as a really little kid. Was going to the Denver Museum of Science, and they had a whole laboratory set up, and you could go in there and you could just take a toothpick and scrape some cheek cells off of the interior of your mouth and put them on a microscope slide. And then if you just looked at the cells directly, you could see something that looked like this. So you can actually see the cell membrane here. You can see that it's enclosed. You can see it's up against some other cells, and you can see there's some stuff inside the cell. But you also can see that the contrast is not so great, right? So this is just looking at these cells directly, and then in this lab, you could just take a little dropper full of dye and drop it onto the sample, wait a few, not very long, just like 30 seconds or so, and then rinse off the excess dye, and then look at it again under the light microscope. And then you saw something that looked like this, where you can see that now we can see a lot more detail about the cell. We can actually see the outline much more clearly of the exterior cell membrane. You can see that it's kind of folded here as the cell is sort of, of course, cell is three-dimensional. It gets crushed onto a two-dimensional slide, so you can see that it's kind of folded up. Now you can see a little bit more detail about some of the things inside, and a very dense region in the center, which is probably the cell nucleus, right, where the nucleic acid is sitting. And so you can see that this stain that was added is very selective for the nucleic acid that's inside of the cell. Okay, now, so light microscopy is great for looking at cells at sort of that level of detail, but what do you want to see things that are a lot smaller? You want to actually see the structure inside of the cell. You want to see some of the organelles inside of the cell. Well, it turns out that then you really need to use a method called electron microscopy, and this was developed in much more recently, around in the 1950s, and scientists realized that instead of using bombarding a sample with light, you can actually use electrons. You can actually bombard samples with electrons, and electrons have a wavelength that's much shorter than the wavelength of light, right? So in principle, one can get much higher resolution by using electrons to interact with a sample and then visualizing the result. And so I want to just focus or mention two different kinds of electron microscopy. One is called scanning electron microscopy, and when you use a scanning EM, what you do is you focus a beam of electrons on the surface of a specimen, just on the surface. And typically what one does is to coat the surface with a very thin layer of a metal like gold, for example. And then the electrons will interact with that gold, and it will reveal a very beautiful three-dimensional structure of the exterior of the sample. And we'll look at a couple of examples of that. And then the other kind of electron microscopy that people use is called transmission electron microscopy. And here, kind of like the name suggests, what one does is to focus the beam of electrons through the sample. So instead of just seeing the exterior architecture of the sample, we actually see inside, and we see the contents of, say, a cell, for example. And these transmission EMs are used mainly to study the internal structure of cells. And so a lot of the information that we have about the organization of cells and what organelles look like and what cell membranes look like and all of that sort of thing comes from studies that have been done using electron microscopy. So let's look at a couple of examples of this. So this is showing a picture that was produced by a scanning electron microscope here of cilia on the surface of a cell. This gives you a sense of the size of the image that we're looking at, the magnification that we're looking at. And so these cilia are you can see they're sort of hair-like structures. They're actually sitting on the surface of the cell, and they're kind of, you get a sense that they have a lot of mobility. They have the ability to kind of wave in the solution, and they're important for cell motility, actually. And so this kind of microscopy then gives scientists a sense of what the sort of big 3D architecture of these kinds of structures, what they actually look like. And then down here, this is showing you what happens if we take this and we just section it, right? We cut a thin section through it, and then we use a transmission electron microscope to look at what's in that thin section. So here, what you can see is that some of these cilia get sliced longitudinally, so we can actually see along the length of the cilium, and some of them get cut horizontally. So then when you cut horizontally, you can see we're looking down a cross-section of the cilium. So we get different sort of complementary kinds of information from these two types of microscopy. And then I also wanted to show you a really cool picture of pollen grains that was produced by a scanning electron microscope. And again, here what you do is to treat the sample typically with a metal such as gold that creates a very thin film over the surface of the sample that will interact with electrons. And I didn't mention, by the way, but one actually uses various kinds of metal containing stains for transmission EM as well, again, to try to increase the contrast of the different parts of the image that you're visualizing. Okay, so again, light microscopy, you want to know, roughly the size range of things that you can look at using it. You want to know that you can use chemical dyes to amplify the contrast, compared to electron microscopy, where we're using electrons and not light to interact with the sample and produce an image. And the size range that we can get down to with electron microscopy is a lot smaller, so we can get to things that are going to be somewhere on the order of, in principle, probably about 1 to 10 nanometers in size. That can be very challenging, but that's sort of in principle where one could get with EM. And we use metals, various kinds of metal-containing solutions to treat the sample to increase the contrast. So hand in hand with microscopy goes biochemistry, as we said. And so this involves actually breaking cells open and fractionating their contents to try to figure out what's inside and what are those molecules and organelles and all the other stuff in the cell, what are those things actually doing. And so it's sort of fundamentally different in a way from microscopy, but because with microscopy we're looking at the whole cell, we're trying to visualize where things inside the cell, and we're trying to look at them in as natural a state as possible. Now, of course, if we add dyes and things, those could, of course, affect the structures, and people have to do very careful controls to check that out. But here's a fundamentally different aspect of experimentation where you're actually, in this case, not even trying to preserve the natural structure of the sample that you're investigating. You're actually just trying to extract certain kinds of either molecules or organelles and understand how they work. Okay, so kind of very complementary sorts of approaches. And very typically, one uses centrifugation to fractionate the cell. So the idea is that you have a sample of cells or tissue, and you use either something like either sonication or a chemical method to disrupt cell membranes. And knowing what you know about cell membranes, and we'll talk about this more later, you can often use things like detergent to break up cell membranes. And then once the cells lyse, they release their contents, and then we can actually centrifuge the sample to fractionate things very roughly by size. Okay, because heavy things will tend to settle and spin to the bottom of a centrifuge tube under that kind of force, and you can use different centrifugal forces to separate things of different sizes. I see a question. Right, so he's asking, would we destroy the cell organelles? And the answer is, it depends on how you do the experiment. Okay, so let's look at the next slide, because this sort of illustrates this. So in principle, what you do here is you actually start off with a sample of cells or tissue. And so this just some things that we work with every day in the laboratory, for example, would be a sample of bacterial cells that might be growing in a flask. But you can also imagine doing this with a sample of liver tissue or some other sort of tissue. And you need some way to break up the cells. And so this shows, if you have a tissue sample, you actually have to literally grind this up in a blender. But if you're working with a sample of bacterial cells, we don't actually have to do that, right? Because the cells are already sort of dispersed in usually a liquid kind of solution. Then we break open the cells either by sonication or by adding some kind of a chemical that will interact with the cell membrane to disrupt it. And then once the cells are lysed, so then we have something that looks like this. They're calling it a homogenate here, but we also call it a lysate, right? A lysate, just something where the cells have released their contents. And then we want to start fractionating. And so the question is, how do we do that without, for example, disrupting organelles? And the way it's done is to just do a sequential series of centrifugation steps in which we start off with a very mild, sort of low-speed spin. And that is too low of a speed to break open things like mitochondria or nuclei if we're very careful. And so very typically you do that very low-speed spin to initially just separate out organelles from the cytoplasm of the cell. Okay, so we do that first, and then we can take those two fractions and work them up further. So if you're trying to prepare cell nuclei, then you might actually be working at that point with what's in the pellet after that first spin. And then you might carefully try to break open the nuclei and let them release their contents. Or if you're working with something that is in the cytoplasm, then you would just take the supernatant from that spin and work with that. And then again, you can continue fractionating by, in each case, spinning the sample with a little bit greater force. And things that are smaller now will fractionate out and pellet to the bottom of the tube. And so this is a way that people were able to do things like fractionate out nuclei, mitochondria, chloroplasts, ribosomes, and then ultimately even smaller things this way. Okay? And so once you have done this kind of fractionation, then you can actually test the activity of, if you're looking for, let's say we're trying to investigate how proteins are synthesized. We would take each fraction that results from an experiment like this and try to figure out which of those fractions has the activity that we're looking for, such as the ability to make a short polypeptide. And that's really the way, fundamentally, that it was originally discovered that ribosomes make proteins, mitochondria are responsible for sort of generating ATP in the cell, and the functions of many of the other organelles as well, what the contents of the nucleus are and things like that. Okay? All right, so I want to just now do just a brief comparison of prokaryotic and eukaryotic cells. And there's a couple things to keep in mind. So first of all, there's some basic features of all cells, right? All cells, doesn't matter what kind of cell it is, have the following things. They all have a plasma membrane that encircles the cell. This is sort of really one of the fundamental things about a cell, right? Is that it has a membrane that separates the interior from the exterior, and it allows chemical reactions to go on in the interior without being unduly influenced by the exterior. Okay? And so this is showing you. Remember last time we talked about phospholipid structure. So this is a really nice picture that kind of shows the lipid bilayer that forms with these phospholipids, where the phosphate groups, which are hydrophilic, are on the exterior of this bilayer, and the hydrophobic tails are interacting with each other on the interior. So inside here tends to be very hydrophobic, and the exteriors tend to be hydrophilic. And then you see these big blobs that are sitting here. Well, it turns out that membranes are not just filled with lipids or made of lipids. They actually include a lot of proteins as well. So there's a number of proteins that are membrane proteins. They sit in the membrane, and they have a lot of very hydrophobic features themselves that allow them to be happy in that kind of environment. All right, so all cells have plasma membrane. All cells also have a cytosol, which is the fluid inside the cell that it's not really, I don't know how fluid it really is, but it's probably more sort of like a sort of gel kind of thing that contains the contents of the interior of the cell. And then all cells have some kind of genetic material, chromosomes that include genes, and all cells have ribosomes to make proteins. Okay, so those are fundamental features that we find in all cells. And then it turns out that we can sort of break cells down further into two fundamental different types. One type of cell is called a prokaryotic cell, and these are cells that correspond to bacteria or archaea. Eukaryotic cells are much bigger cells that have a nucleus, and these are cells that correspond to fungi, protists, plants, and animals. Okay, so those are two sort of fundamentally different kinds of cells that we find in biology, and let's have a quick look at what those look like. So this is a slide from the book that shows the basic structure of a bacterial cell. You can see these cells are often rod-shaped. There's no nucleus in a prokaryotic cell because the DNA in these cells is actually it's not just floating around randomly. It's actually in a sort of localized region called a nucleoid, and there are proteins that help package that DNA, but it's not in a membrane-enclosed structure like a nucleus in a prokaryotic cell. And we don't see membrane-bound organelles inside of a bacterial cell, and we also don't see we see that the cytoplasm is bound by the plasma membrane. Okay, and you can see there's also some other things going on here. There's lots of these little cilia. There's flagella that help the cell to move around, and then here's an actual transmission EM micrograph that shows you what this would look like if we sliced through it and actually just looked at it with an electron microscope. Okay, so bacterial cells inherently are a lot smaller than eukaryotic cells. And here's a picture, again, a cartoon drawing of a eukaryotic cell. And what you see here is that the DNA is now located in a nucleus. So this is bound by what's called the nuclear envelope. And the cool thing about the nucleus is that it's actually a double membrane, right? It's not a single